This video lecture covers the ABO system for blood types using Punnett Squares to determine possible blood genotypes for offspring. It is recommended that this lesson be used in conjunction with a lesson on Punnett Squares and genotype configurations. Rebecca Bennett is the original creator, and modifications were done by Derek Smetzer. This work was made possible through the NSF-funded Photo Knowledge in the Science Classroom project at Ohio University. The ABO system is a blood type classification system based on 4 basic blood types: Type A, Type B, Type AB, and Type O. You may already be familiar with some of the specifics of this system if you have ever donated or received blood before. Each blood type is distinguished from the other by the inclusion of a unique carbohydrate group attached to a protein on the surface of the red blood cells. The specific structure of this protein will determine what blood type you happen to have. The image is a crude simplification of the proteins on the surface of each red blood cell type. There are many proteins on the surface of a blood cell, but the proteins of most importance are those corresponding to blood type. We will find out why later, but first it's necessary to understand a little bit about the structure of these proteins. The carbohydrate portion at the end of the protein is depicted above for type O blood cells. This terminal carbohydrate structure is shared among the proteins of the other blood types. For both type A and type B, an additional sugar molecule is attached at the furthest end of the carbohydrate. In type A, this is an N-acetylgalactosamine group, and in type B, a galactose group. Type AB blood is unique in that it has both the A and B proteins attached to the surface. The surface proteins corresponding to blood type are known as antigens. Type A blood cells have the A antigen, type B blood cells have the B antigen, type AB blood has both A and B antigens, and type O has the H antigen. The H antigen is the precursor antigen to the other blood type antigens. As we previously learned, the surface of red blood cells are covered with unique proteins called antigens. Antigens trigger the introduction of antibodies into the immune system to fight off infection and foreign bacteria. In the presence of an incompatible blood type, antigens will trigger the introduction of antibodies, which destroy the foreign blood cells. Normally, our immune systems don't produce antibodies against the antigens that are part of our blood. Autoimmune disorders occur when the body's immune system sends antibodies to attack healthy cells marked by our own antigens. Given the information from before, see if you can come up with the answers to this table. Pause the video here and write which antibody is present in each blood type. If you have type A blood, you have A antigens and anti-B antibodies in your blood. Similarly, if you have type B blood, you have B antigens and anti-A antibodies. So people with type A blood will reject a blood transfusion from a person with type B blood and vice versa. In the case of type AB blood, you have both A and B antigens and no antibodies in your blood. People with type O blood have neither A or B antigens, but have both anti-A and B antibodies. Type AB blood is considered as a universal acceptor because there are no antibodies for A or B antigens. Type O is the universal donor because it has no A or B antigens and cannot be attacked by anti-A or B antibodies. However, you cannot donate AB blood to people with A, B, or O blood types, and if you have type O blood, you cannot receive blood from anyone other than a type O. This is why blood transfusions can be dangerous. If you are given the wrong type of blood, the antigens and antibodies in your blood can cause a harmful reaction, leading to red blood cells bursting or clumping together, causing blocked blood vessels. The blood type gene has 3 different alleles. Capital I with an A superscript corresponds to the A allele, capital I with a B superscript corresponds to the B allele, and a lowercase I corresponds to the O allele. Both the A and B allele are dominant traits, while the O allele is recessive. How many genotypes can be made from these alleles? How many phenotypes exist? Pause the video here and try to answer this question. There are 6 possible genotypes associated with these alleles, and 4 phenotypes result from these genotypes. The possible genotypes are homozygous dominant A, heterozygous AB, heterozygous A, homozygous dominant B, heterozygous B, and homozygous recessive O. The phenotypes which result are simply the four blood types associated with the ABO system: A, B, AB, and O. The Rhesus blood system is an extension to the ABO system that also considers an Rh antigen. You may be most familiar with this system: A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative are all the possible phenotypes when including the Rh factor. An Rh positive corresponds to the presence of the Rh antigen, while an Rh negative means that the antigen is not present. In this system, the universal donor is O negative. There are no antigens and therefore no antibody response. The immune system does not detect O negative as foreign in this case. The universal acceptor is AB positive. AB positive has A, B, and Rh antigens and produces no antibodies against A, B, or Rh antigens. There is no immune response against foreign blood cells of any type. Using the information you've just learned about blood types, try using a Punnett square to determine the possible genotype for a child with a mother having type O blood and a father having type AB blood. Pause the video here and resume once everyone has found an answer. In this case, the mother is homozygous recessive because type O blood is a recessive trait. We denote this by writing two lowercase i's above the Punnett square in each column. The father is heterozygous since type AB blood carries an A and B allele. This is denoted as IA and IB corresponding to each row. The genotype for the child results in a 50% chance of heterozygous type A blood and a 50% chance of heterozygous type B blood. Therefore, the possible phenotypes for this child are either type A blood or type B blood.